Welcome everyone to the third video of this series, and today we are going to learn about the diseases that fall under hemolytic anemia. There will be a part 1 and part 2 of this video. So without any delay, let's start with the topic. Let's first start with why anemia happens due to G6PD deficiency. So we can see the pentose phosphate pathway in the left hand side, and pentose phosphate pathway is also known as HMP shunt pathway. In this pathway, glucose 6-phosphate is oxidized by glucose 6-phosphate dehydrogenase to form 6-phosphogluconolactone. In this pathway, we get NADPH in various steps, which is the reduced form of NADP. And this NADPH is an electron donor. Now let's learn about how NADPH helps in the protective mechanism against free radicals. Okay, so whenever there is any kind of oxidative stress, reactive oxygen species or free radicals are formed. For example, superoxide, hydroxyl radical, etc. It can come from mitochondrial respiration, or it can be formed due to inflammation in your body, or there can be drug metabolism, toxins, radiations which can form this reactive oxygen species. Now, free radicals can assist the process of lipid peroxidation. Lipid is present in the membrane, so there can be membrane rupture, membrane damage, and the cell can rupture. It can also lead to protein damage, so the hemoglobin present in it gets damaged. But RBC has an antioxidant defense system. What is this defense system known as? This defense system is called glutathione system. Now, what does this system do? This system has a hero. The hero is glutathione peroxidase. And also this system has a villain, which is hydrogen peroxide, H2O2, and it acts as a toxic substance. Now, the hero converts the villain into a less toxic form. So, hydrogen peroxide is converted to water, which is safe. But in this process, the reduced form of this enzyme is converted into the oxidized form. But we need to reuse it again, right? So we have to convert the oxidized form back to the reduced form. And we do this with the help of another enzyme known as glutathione reductase. You can remember it like a story where the hero tries to save the world and converts the villain into a less toxic form. But in this process, he loses all his power. But now the heroine, which is glutathione reductase, has healing powers. So she heals the hero. But this heroine needs a weapon. And what is the weapon? The weapon is NADPH. In this process, NADPH donates a proton to the oxidized form of glutathione to convert it into the reduced form, and itself gets converted into the oxidized form, that is NADP. It loses its hydrogen. And from where do we get this NADPH? We get it from the HMP shunt. And that's why glucose 6-phosphate dehydrogenase enzyme, that is G6PD enzyme, is so important because it helps to protect the cell. Now, let's learn more about this G6PD deficiency. It is an X-linked trait. Hence, males are at a higher risk than women because males have only one X chromosome. Different mutations can produce variations like G6PD-A and G6PD Mediterranean. This G6PD-A is milder and it is found in 10% of black Americans. However, G6PD Mediterranean is more severe and it is found in the Middle East countries. Now, one of the characteristic features are episodic hemolysis, which is the sudden destruction of RBCs that occurs only when triggered by stressors like infection, certain drugs, or oxidative stress. So what are the triggers? The triggers can be infections like viral hepatitis, or it can be pneumonia, typhoid fever, etc. Drugs like anti-malarials, for example, primaquine, chloroquine, sulfonamide, etc. Also very commonly, the intake of fava beans, which contain oxidant compounds, generate oxidative stress in the RBCs. Now what happens when there is G6PD deficiency? When there is no G6PD, reactive oxygen species are not neutralized. So they react and leads to the cross-linking of sulfhydryl groups on the globine chain. This leads to their denaturation and they form membrane-bound precipitates, which are called Heinz bodies. It is seen as purple-blue inclusions after we apply stains like methyl blue. and these purple-blue inclusions are composed of denatured precipitated hemoglobin. These precipitates can damage the membrane sufficiently to cause intravascular hemolysis. Also, when this RBC passes through liver or spleen, macrophage removes the Heinz body containing RBC. What does it do? It takes out a bite of the damaged area only and forms a cell which has a half-bitten area. These are known as bite cells. Now let's look at the microscopic picture of anemia due to G6PD deficiency. Can you locate the precipitates in the cell? The blue ones? Yes, these are the Heinz bodies. You can also locate the half-chewed cells, and these are the bite cells where the macrophages have taken a bite out of the cell. Now what happens to the less severely damaged cells? They become spherocytes, and both the spherocytes and bite cells are removed by the phagocytosis process. Hence, we can see both extravascular hemolysis and intravascular hemolysis. Now, there can be two types of screening tests to find it out. One, methemoglobin reduction test, and two, fluorescence spot test. In the first one, we take a blood sample containing hemoglobin and we add sodium nitrite. This sodium nitrite will convert hemoglobin to methemoglobin, and there will be a change in the color from red to brown. Now, if we add methylene blue, this will activate the G6PD pathway. If G6PD is present in the blood, then this brown color will convert back into red color because methemoglobin is converted back to hemoglobin. But if G6PD is deficient, then there will be no color change. Now let's move on to fluorescence spot test. NADPH is autofluorescent. So if G6PD is present, then NADPH is formed, and under UV light, a fluorescent glow is seen. If there is no fluorescent, then we have to understand that G6PD is deficient. And for the confirmatory test, we do a G6PD assay, which is basically a kind of ELISA test. So that was all about G6PD deficiency, and now we will move on to hereditary spherocytosis. Hereditary spherocytosis is a genetic disease which can either be autosomal dominant, which happens mostly, or it can be autosomal recessive, which happens in 25%. Now, it is caused by intrinsic defects in the red cell membrane skeleton, which makes the red cell spheroid, less deformable, and vulnerable to splenic destruction. Now, let's see what the membrane skeleton is made up of. First and foremost, we can see spectrin, which is formed of two polypeptide chains, alpha and beta, which are intertwined with each other, and they form heterodimers. Next, we can see ankyrin, band 4.2 and band 3 on the same side. Now, this ankyrin and band 4.2 binds spectrin to band 3, and band 3 is a transmembrane ion transporter. It helps to transport ion. You can see the same in the diagram how ankyrin and band 4.2 connect spectrin to band 3. On the other side, we have actin, then we have band 4.1, and we have GP, which is glycophorin A. Now here also, actin and band 4.1 helps to bind spectrin to glycophorin A, which is a transmembrane protein. Now a few points to be noted. The biconcave shape of RBC is due to spectrin. The most common mutation happens in ankyrin in hereditary spherocytosis, and least common mutation occurs in glycophorin A. Now let's learn about the pathogenesis. So there can be various mutations. These mutations can lead to the insufficiency of membrane skeletal components that we just read about. This decreases the lifespan of an RBC from 120 days to just 20 days. What happens is they destabilize the overlying plasma membrane, right? Because the membrane skeletal components are insufficient. And as the RBC becomes old, it sheds membrane fragments. This forces the cell to assume the smallest possible diameter to become stable, and the smallest possible diameter is acquired by a sphere. Now let's see this diagram from Robbins. Here we can see that the plasma membrane has been destabilized. Next, what happens? It sheds its membrane fragments, and after that it becomes a sphere, which is the smallest possible diameter, and it becomes a spherocyte. Now there's a question why removal of spleen is beneficial in this case. See, red cells must undergo extreme deformation to leave the splenic red pulp cords to enter the sinusoids in the spleen. But spherocytes are not so flexible, so they get trapped in the cords and they become a prey to the macrophages. Also, there is high tendency of spherocytes to lose membrane. Hence, potassium and water escapes and it becomes dehydrated. Hence, in this case, splenectomy is beneficial so that spherocytes do not have to go through all these traumas and they will still persist, but anemia will be corrected as they will not die. Now, let's look at the clinical features. Normally, there will be spherocytosis that can be seen. There can be reticulocytosis, which is highly suggestive of hereditary spherocytosis, and there can be also negative direct Coombs test. Reticulocytosis happens because when there is anemia, the bone marrow tries to compensate and makes more RBC, and reticulocytes are the precursors of RBC, right? And Coombs test tells us if there is antibody or complement or RBC, which is not there in case of hereditary spherocytosis. Hence, we get it negative. Also, due to membrane rupture, there is loss of potassium and H2O, which leads to dehydration. And in dehydration, we have read that mean corpuscular hemoglobin concentration is increased because hemoglobin is more concentrated when there is dehydration. We can also use a fluorescent dye which is known as eosin 5 malamide, which binds to band 3, and in this case staining is decreased in red cell disorders like hereditary spherocytosis because of loss of membrane structures. As extravascular hemolysis happens, hence the clinical features will be anemia, splenomegaly and jaundice. So what are the types of crisis that we can see? So first of all, we can see aplastic crisis and we can see hemolytic crisis. What is aplastic crisis? It is a sudden halt in red blood cell production by the bone marrow, which is usually triggered by parvovirus B19. And hemolytic crisis is caused by infectious mononucleosis, where we have severe splenic destruction of RBC. Also, we can sometimes see gallstones in this case. Now let's look at the morphology and lab findings. So under the microscope, we can see circular cells called spherocytes, and they are darkly stained, which means they are hyperchromic. They lack the zone of central pallor. Okay, so they are completely filled. And then there is variation in sizes. So we can see anisocytosis. Then there is reticulocytosis because of the compensation by the bone marrow. So we can also see erythroid hyperplasia. Other features are hemosiderosis, mild jaundice, cholelithiasis, which are pigment stones, and splenomegaly. So this was all about hereditary spherocytosis, and now we will move on to paroxysmal nocturnal hemoglobinuria. Now this disease is not genetic, it is an acquired mutation. Where does this mutation happens? It happens in a gene called PIGA gene. The full form of PIGA is phosphatidyl inositol glycan complementation group A gene. You do not have to learn the full form, just pick up the word complementation group. Okay, this PIGA gene makes certain membrane associated complement regulatory proteins. So we can understand that it regulates the complement system. Now let's look at the diagram to understand better. This PIGA gene makes an anchor called GPI, which is glycosyl phosphatidyl inositol. Some proteins are attached to the cell membrane through this GPI anchor. Now look at the diagram. This is the GPI which is connecting the protein to the cell membrane. Also remember, this is an X-linked disease and the mutations occur in the hematopoietic stem cell. Hence, RBC, WBC, platelet, all are affected. All are deficient in GPI-linked proteins. Now, what are the GPI-linked proteins that are affected? Firstly, CD55 is affected, which is also known as decay accelerating factor, DAF. Next, CD59 is affected, which is the membrane inhibitor of reactive lysis, and also C8 binding protein is affected. CD55 inhibits C3 convertase, which is a part of complement system. CD59 inhibits MAC, which is the membrane attack complex. Hence, all these proteins help in the inhibition of the complement system. Therefore, if these proteins cannot exist due to the deficiency of the anchor, what happens? there is activation of the complement system, and due to this activation there will be lysis of the RBC, which will be intravascular. Now, why do we have the name paroxysmal nocturnal hemoglobinuria? Because at night our respiratory rate is very low, hence there is accumulation of CO2 and there is acidosis. pH is decreased, which is good for the complement system, and hence it activates the complement system, which leads to destruction of the RBCs, for which hemoglobin is released and we get hemoglobinuria, which means hemoglobin is found in the urine. Now, let's look at the clinical features. We already know there will be hemolytic anemia, which will be intravascular. Next, there will be hemoglobinuria, which means we will find dark urine at the morning. Then we will have pancytopenia, which means there is a reduction in all three major types of blood cells in our body. Next, there can be thrombosis, which is the leading cause of death in PNH. Thrombosis can happen in hepatic veins, portal veins, and in cerebral veins. Now, why do thrombosis happen? Because the free hemoglobin that is there in the blood binds to nitric oxide. This nitric oxide is a vasodilator. It prevents the clumping of the platelets. But if free hemoglobin binds to nitric oxide, then it cannot do its job. Hence, there is a higher risk of thrombosis. Also, there can be endothelial damage due to membrane attack complex of the complement system. There can be bone marrow cancers like acute myeloid leukemia or myelodysplastic neoplasm. Now, what are the screening tests? There can be two types of tests. One is HAM test and the other is sucrose lysis test. In HAM test, we provide an acidic pH. We acidify the medium. Okay, an acidified medium will be activating the complement. Now, if it is a normal RBC, then it will have the complement inhibiting proteins. So there will be no lysis. But if the person is suffering from PNH, then the RBC will do not have the complement inhibiting proteins and it will be destroyed. So hemolysis will be observed. Now what happens in the sucrose lysis test? In sucrose lysis test, the sucrose reduces the pH. It will also activate the complement system, and hence in normal RBCs, there will be no lysis, but in the affected RBCs, there will be lysis. So if hemolysis is observed, sucrose lysis test will be positive. Now how it is confirmed? It is confirmed by flow cytometry. It detects the RBC that is deficient in GPI linked proteins like CD59. So that was all about paroxysmal nocturnal hemoglobinuria. Now you can go and read the same topic from Robbins. Today I have covered hereditary spherocytosis, G6PD deficiency, and paroxysmal nocturnal hemoglobinuria. In the next class, we will be covering the rest of the diseases under hemolytic anemias.